In 1858, Carl Linnaeus assigned the domestic dog its scientific name, placing it in a single category alongside the wolf and the jackal, without controversy. At the time, nobody had reason to think that classification would ever be tested. A dog was a dog. It would be more than 200 years before anyone had the tools to ask whether that single label could actually hold. Today, it's tested constantly, and mostly by accident, inside veterinary clinics and breeding kennels around the world every day. A Mastiff weighing over 200 pounds and a Chihuahua weighing under 6 both carry the same species designation on their paperwork.
Measured by skull proportions, the method taxonomists use to distinguish wild carnivore species, the shape difference between these two dogs is extreme. That shape difference exceeds what typically separates entire genera within Carnivora, the taxonomic group that includes wolves, bears, and seals. Under a microscope, their chromosomes tell a different story. 39 pairs each, pairing off with no resistance or mismatch, as within a single litter. That contradiction is not a footnote in the history of the dog. It is close to the center of it. A body shaped this freely, sitting on top of a genome that barely moved, is not something evolutionary biology predicts easily. Reconsidering those two facts, a freely shaped body and a nearly unchanged genome, is where this documentary is spending its time. The question of whether the term same species still genuinely applies to today's dogs is not merely rhetorical. It has a specific, testable answer, and biologists have spent several decades carefully building the scientific case for it. The case doesn't rest on appearance, because appearance is precisely what 2 centuries of selective breeding have been rearranging. Coat, height, and skull shape have all been actively pushed toward extremes, which disqualifies them as evidence one way or the other. What biologists rely on instead is the machinery underneath: chromosome number and reproductive compatibility, traits breeding has never had a direct route to alter. Every domestic dog ever examined carries 78 chromosomes, arranged neatly into 39 matched pairs, regardless of the animal's size. When reproductive cells from dogs at opposite extremes are compared under a microscope, the pairing process runs cleanly, as it would within a single breed. There is no genetic evidence, at that chromosomal level, of 2 lineages that have meaningfully drifted apart over time. By the standard definition of a species, shared chromosome architecture, and full reproductive compatibility, the smallest and largest dogs on record remain, unambiguously, one animal. That finding sets up the real subject of this film: a species capable of holding this much external variation while its genetic core stays fixed. Such a pattern is unusual enough to demand an explanation, and the search starts with a more basic question. How much time did this take? Through most of the 20th century, the working assumption was that dog diversity had built slowly since domestication, a process spanning roughly 15,000 years. Under that assumption, wolves gradually edged toward each modern breed shape over dozens upon dozens of successive generations. That long-held assumption turns out to be largely wrong, and the historical record makes the case quite cleanly. Breed stud books, kennel club records, and the archaeological and artistic record of dogs across the preceding millennia point to something different. For nearly all of that 15,000-year span, dogs varied within a narrow range, recognizably dog-shaped, and descended from wolves, without anything resembling a modern pug. The dramatic divergence that produced most of today's recognized breeds is concentrated entirely in the last 200 years. This divergence correlates tightly with the rise of organized dog showing and formal breed standards in Victorian Britain. Historian Michael Warboys and colleagues have documented how this happened. A relatively small circle of 19th century breeders operated through newly formalized stud books. Through competitive show rings, these breeders converted loose regional dog types into fixed, codified breeds within just a handful of generations. The dogs most people picture when they think of breeds today are not artifacts of deep prehistory at all.
They are, on an evolutionary scale, essentially contemporary, closer to a few dozen human generations removed from us than to the earliest domesticated wolves. That timeline raises a genuine biological problem, because morphological change of this magnitude does not typically occur this quickly in mammalian populations. The resolution turns out to be genetic rather than historical, a small number of genes, each with an outsized effect on body shape. That small number of genes made 2 centuries long enough to accomplish work that would otherwise have taken far longer to complete. The initial expectation going into this research was that dog body size would work the way human height does. Human height is a trait controlled by thousands of genetic variants, each contributing a small, largely interchangeable effect that adds up gradually across a population. If that model held for dogs, breeders would have needed an enormous number of generations to reshape body size and proportion this dramatically. This is because polygenic traits of that kind tend to shift slowly across a population, one small genetic increment at a time. Genome-wide comparisons between giant and toy breeds, however, didn't support that slow, thousands of variants model of dog body size. Rather than thousands of small contributors, researchers led by Nathan Sutter and later by Adam Boyko found something strikingly different in the genome. A small number of genetic loci account for most of the variation in body size, leg length, and skull proportion across dog breeds. These are differences substantial enough to separate other mammal species by millions of years of divergence, concentrated in just a handful of genome locations. One variant near the IGF-1 gene alone explains a large share of the size difference between small and large dog breeds. This is a fundamentally different genetic architecture than the one governing human height, and it explains the speed of the Victorian transformation directly. With only a few high-effect loci controlling body shape, selecting hard for specific variants produces dramatic change within a few dozen generations, rather than hundreds. What this architecture does not explain on its own is a separate, more puzzling pattern. Traits with no obvious genetic connection to each other. Traits like ear shape, coat pigmentation, and temperament keep appearing together across unrelated dog breeds and unrelated domesticated species entirely. That clustering problem predates modern genetics. Charles Darwin described it in 1868, noting that domesticated animals across different species shared a consistent set of traits. These shared traits included floppy ears, patches of depigmented fur, and reduced fear of humans, despite having no obvious biological reason to be connected. Darwin documented the pattern without being able to explain its cause, and for more than a century afterward, neither could anyone else. Dmitry Belyav's fox domestication experiment began in Soviet Russia in the late 1950s, continued for decades by Ludmila Trut, selecting foxes purely for tameness. Within a small number of generations, those foxes began developing floppy ears, curled tails, and piebald coats that no one had selected for directly. The clustering isn't specific to dogs. Similar clustering has since been documented in farmed mink, laboratory rats, and cattle as well. The explanation gaining the most traction among developmental biologists, proposed by Wilkins, Wrangham, and Fitch in 2014, traces the pattern to the neural crest. These cells form early in development and migrate outward across the embryo towards several different destinations at once, all simultaneously. Their destinations include the developing face, the skin's pigment cells, the forming ear cartilage, and the adrenal glands, which regulate stress and fear. Because these structures derive from the same starting population of cells, a change affecting neural crest behavior early in development can influence several traits. That single change can influence pigmentation, ear structure, and fear response simultaneously, even though those traits appear entirely unrelated in the adult animal. Under this model, selecting an animal for reduced fear of humans is, at the cellular level, indirectly selecting for a change in neural crest activity. Because that same cell population also builds the ear cartilage and skin pigment cells, floppy ears and coat patches can appear as a side effect.
That side effect appears when selecting purely for calmness, without those floppy ears or coat patches ever being selected for directly by anyone. The hypothesis remains the leading explanation in the field, though researchers have noted it doesn't account cleanly for every domestication-associated trait in every species. It does, however, clearly explain why the same combination of features keeps reappearing across many completely unrelated domesticated animal species. That leaves an unresolved gap. This genetic and developmental machinery has, by all indications, always existed in wild mammal populations everywhere. Yet wild wolf populations have never independently produced anything resembling a pug, a Chihuahua, or a Mastiff on their own. The explanation for that gap is not genetic, it is environmental, and comes down to what a wild population's surroundings allow to survive. The answer has less to do with genetics than with the environment those genetics would actually have to survive in. Consider what each of these extreme body types would face outside of a human household in a truly wild setting. A bulldog-type skull, with its shortened airway, struggles to move enough air to cool the body efficiently in a wild setting. That becomes a serious liability during sustained heat or exertion, particularly when a bulldog-type dog is evading a predator. A body at the other extreme, small enough to match a Chihuahua, has a correspondingly high surface area to mass ratio, losing heat quickly. That body loses heat faster than its metabolism can replace it, making an unsheltered winter night a serious survival risk. A body at the largest extreme, closer to a Mastiff, requires substantially more food to sustain its considerable mass in the wild. In a wild population without a guaranteed food supply, that large body size becomes a liability during any prolonged food shortage. Each of these represents a different mechanism, but the underlying principle is the same across all three extreme body types. The genetic switches capable of producing these body types have likely always existed in wolf populations, arising the same way any mutation does. What has consistently been missing is simply the opportunity for those mutations to persist long enough to actually spread. In a wild population, every generation passes through a filter that removes individuals whose bodies can't sustain themselves, regardless of the mutation's origin. Population geneticists call this stabilizing selection, a pressure that doesn't generate new traits, but continuously removes extreme variants from a population. This keeps the average body plan within a survivable range across every successive generation of a wild wolf population. Domestication altered the criteria that filter operates on entirely, changing what determines an individual dog's survival and reproduction. Removed from a wild environment with stable food, shelter from extremes, and no predation, a dog's body no longer needs to be self-sustaining. Survival and reproduction now depend instead on human preference, whether a breeder favors a particular skull shape, size, or coat pattern. That shift didn't create new mutations or a new genetic mechanism. It removed the selective pressure, eliminating variants for as long as wolves have existed. This allowed genetic switches that nature had always possessed to be pushed further than natural selection alone would ever have permitted. Removing that pressure did not remove its consequences. It simply relocated them, from population-level survival to individual-level health, as three breeds illustrate clearly. In Chinese Shar-Peis, researchers led by Mia Olsen identified a genetic duplication upstream of the HACE2 gene responsible for the breed's characteristic skin folds. That same duplication increases hyaluronic acid production throughout the body, which also predisposes the breed to a hereditary condition known as periodic fever syndrome. This periodic fever syndrome causes recurring, unexplained fevers in Shar-Peis, with no infectious cause behind them at all. The trait that defines the breed and the disease associated with it originate from the same genetic change. In Rhodesian Ridgebacks, the breed's signature dorsal stripe of reversed fur is associated with a different DNA duplication affecting skin development. That genetic region has been linked to an elevated risk of dermoid sinus, a defect where the neural tube fails to separate from the skin. As with the Shar-Pei, the visible breed trait and its associated health risk trace back to the same underlying mutation. In brachycephalic breeds, like Bulldogs and Pugs, selection for a flatter, infant-like facial profile compressed the skull faster than soft tissue could adapt. This mismatch produces the breathing difficulty now common across these breeds, a well-documented condition known as brachycephalic obstructive airway syndrome. The cost of 2 centuries of intensive selection extends beyond individual traits into the genetic structure of entire breed populations.
Popular sire effects occur when a single, heavily used stud produces an outsized share of a breed's next generation, sometimes numbering in the thousands. These popular sire effects have measurably narrowed genetic diversity within closed breed populations over many successive breeding generations. Genomic studies estimate that the average purebred dog carries an inbreeding coefficient comparable to that of human full siblings, though this varies by breed. Multiple dog breeds show substantial losses of genetic diversity, specifically within immune system genes responsible for recognizing and fighting pathogens.
Despite 2 centuries of selection focused heavily on temperament and behavior, a 2022 genomic study led by Kathleen Morrill and colleagues examined thousands of dogs. This study found that breed identity accounts for only a small fraction, roughly 9%, of the variation in individual dog behavior. Most behavioral traits are highly heritable within individual dogs, but they are not reliably predicted by which breed category a dog belongs to. This means the behavioral stereotypes commonly associated with specific breeds are far less consistent than popular assumption generally suggests.
Taken together, the evidence points to a fairly specific conclusion about where this genetic capacity for variation actually originated. The genetic capacity to produce this range of body types was not created by selective breeding. It already existed within wolf populations. It exists in the form of a small number of high-effect genetic loci and a shared developmental pathway running through the neural crest. What selective breeding changed was not the raw material, but the filter, removing for roughly 200 years the survival pressures that constrained those variants.
That removal is reversible, and free-breeding dog populations demonstrate it directly across generations without any human-imposed pairing at all. Village dogs and similar free-breeding populations consistently converge on a similar general body plan, medium-sized, short-coated, with balanced proportions. When the selective pressure exerted by breeders is removed, the extremes recede first, and the ancestral default reasserts itself. There is one additional finding worth noting. The SMOC2 gene, implicated in the facial compression seen in pugs and related breeds, matters elsewhere too. Researchers studying human craniofacial development have identified SMOC2 as a candidate gene of interest in certain birth defects affecting skull and facial formation. The same genetic mechanism responsible for a health cost in one species is now informing medical research in another, entirely different species. This is not a justification for that cost, but a demonstration of how the same biology can produce very different outcomes depending on context. What this body of research ultimately shows is not that dogs are a biological anomaly, but that these genetic tools were ordinary all along. What changed for roughly 2 centuries was simply which pressures were allowed to act on those ordinary genetic tools.